Okay, so let's get started on time. So just in case we run over. So once again, hopefully more groups will show up because we're supposed to have six groups. So be prepared to go out of order if the next group isn't here in time. So the first group is group four, and basically just like last time, I will set the timer for 10 minutes and 30 seconds just so you have a sense when it goes off about that you should be wrapping things up if you haven't already wrapped up. If you're done before, then obviously that's fine too. Um, just remember to let's see. I'll mute and then you guys unmute. Okay. So I'm gonna mute now.
Hi guys, we're group four, and we'll be talking about some methods for computing absorbed radiation dose in CT imaging. Let's start by looking at the three main types of doses that are seen in CT imaging. We start off with the absorbed dose, which is measured in gray or GY, and that is a fundamental measure which tells us the amount of energy that is deposited in a specific tissue volume. Now, this can be converted into equivalent dose by incorporating a radiation factor, which is called WR, and that just tells us how sensitive a particular kind of tissue is to a particular kind of radiation. And this moves on into effective dose, which is in Sieverts or SV, which then takes into account the specific composition of a person, because different tissues can actually be impacted in different ways. And you can kind of see that in a diagram where maybe something like a thyroid or a lung is more sensitive to radiation than other parts of the body. Now, why does dose estimation matter at all? It's a really crucial first step in any kind of clinical practice when you're doing a CT scan on a person, because there are two kinds of important radiation exposure factors. We have deterministic effects, which can occur usually at high doses, and there's a threshold below which it doesn't occur. And they're usually more superficial in nature. The more dangerous ones are stochastic effects, which can occur at any dose, and there is no threshold for that. They're random in nature, and they also have much more serious consequences, you can see in the form of cancer or genetic mutations. Now, if we look at the example of trying to mitigate damage from radiation dose in a vulnerable population like pregnant women, we can see that's really crucial because it not modulating fetal CT dose can actually lead to increased risk in childhood cancer, mutations within the fetus itself, and developmental issues. So the general aim is always to keep the radiation dose ALARA or as low as reasonably achievable. And the first step in this is accurate dose estimation. Starting off with some dose metrics which are commonly used in a clinical setting, we have CTDI or the CT dose index, which is just the which is just a measurement of the dose across a single slice in a CT. And this can be divided into CTDI 100, which again is just the dose at the center of the slice, which can be converted into CTDI W by weighing it based on the fact that the periphery gets a lot more of the dose than the actual center. And if we adjust for pitch in stuff like helical scans and multi-slice scans, we actually get CTDI volume. If we convert CTDI volume into DLP, which is dose length product, we do that by multiplying it by the entire region that scans. We can see that a CTDI volume multiplied by the entire scan length, and this gives us a total measure of the radiation that has been deposited onto the entire scanned area. Now, while clinical dose metrics are very common and have been widely adopted, there are some limitations that they have, which we can use Monte Carlo simulations to overcome. And some of these that we've listed here are lack of personalization across patients, because CTDI and DLP are generally standardized metrics, which have been calculated based on average patient models. So they don't account for any variation. And this means that if your patient's body differs from the norm, that can cause some problems. Monte Carlo simulations can actually take into account specific tissue or anatomical variations and help us do patient-specific modeling. Another problem with clinical metrics is the fact that they oversimplify dose distribution because they don't take into account dose distribution depth or any kind of probability distribution with the dose. This can be modified with Monte Carlo simulations, which is a probabilistic simulation method and takes all these parameters into account. And finally, there's a limited relevance for risk prediction with dose metrics because they can't be there's no longitudinal study for these. They can't be measured over time. But Monte Carlo simulations can be repeated time and again and can be combined to kind of figure out a cumulative risk profile for the patient.
So how are Monte Carlo simulations with dose estimation? We categorize this into three main steps that you would do. The first step is doing the photon generation. So you need to generate the photon energies using the cumulative distribution function, the CDF of the X-ray energies. And this equation looks like the energy of the photon is equal to the inverse of your CDF with respect to R, and R is a number from 0 to 1, which is your probability. Step two would be simulating the photon transport. So how are photons moving in the body? And we can look at this by looking at the attenuation model, which describes the intensity of the photon beam decreasing as it travels through the tissue. And so your attenuation coefficient is mu, and then your initial intensity before the photon beam hits the tissue, and then the intensity after. Third step is calculating the dose. So the energy transferred to the tissue per unit mass. There are some key scanner parameters and their relation to dose that we'll go over. The first parameter is pitch. Pitch is how fast is the table moving with respect to the X-ray beam. This ends up being inversely proportional to the dose. Second parameter is rotation time. So what is the time taken for the X-ray detectors and the X-ray tube and the detectors to complete a full rotation around the patient? This ends up being directly proportional to dose as well. Third parameter is the X-ray tube voltage. So this determines how much energy do your photons have. This is also directly proportional to dose. And the last is the source to image distance, your SID. So this is the distance between the X-ray tube to the detector. And this is related by the inverse square law to the dose. Next, we're going to look at what are some dose modulation and reduction in CT scans. First is the tube current modulation, which you can see in the figure. And what the tube current modulation does is it will change the tube current based on the patient's anatomy. So regions that are more dense will receive more current, as opposed to regions that are less dense, they'll get less current. And this is a balance between image quality and dose. The second is we have physical filters. So figure 2, you can see an image of bismuth shielding, and they'll put this filter on more sensitive areas where you don't want to get bombarded with X-rays, such as like the breast, thyroid, or eye. And then figure 3, we have a bow tie filter, and you can see it's in the shape of the beam. So like you get most of the X-ray energy photons going to the centric part of the body, and then the periphery of the body gets less because we don't need dose there. So we wanted to do a Monte Carlo simulation of our own. We ended up finding a database where that group actually did their own Monte Carlo simulation. So we had that as our ground truth. And the database included 24 women that were pregnant, and we had ranging gestation periods from 5 to 36 weeks. And this data set gave us information about the scanner itself, the patient information, exam specifications, and their own Monte Carlo simulation parameters. For our simulation, we assumed the same helical CT scanner. We had a rotation time of 0.5 seconds, a pitch equal to 1, and we assumed a 19 millimeter nominal beam collimation.
So for our data set, we also had a mapping between the tissue densities and the voxels. So we used that to create this segmented voxelized model. You can see the 3 different views here: coronal, axial, and sagittal for different tissues, color coded in different colors. So to give a brief about our MC simulations, inherently these simulations are very complex and computationally intensive. So we implemented a lot of approximation to reduce the computation time. For example, we did the slice-based dosage estimation for one specific fetus model. We downsampled this slice image to almost 1/4, and the cumulative distribution of the photon energies were already given from the data set. For example, this is the CDF for 120 kilovolt tube voltage. And we did simulation only for FTC, that is fixed tube current, because TCM is even more difficult to simulate. So in total, we simulated 100 photons per 10 degree of angle per voxel. And out of these 100 photons, we calculated the attenuation based on the source and the distance between the source and the voxel. We calculated this distance based on the image position or orientation from the DICOM images and SID from the scanner. And we scaled this by factor of the equivalent diameter of equivalent water diameter. After that, we calculated the probability of interaction of these remaining photons with the tissue based on energy of the photons and linear attenuation coefficient. We scaled this attenuation coefficient by factor of 3 to incorporate our approximations. And in the end, whatever photons number we had for particular voxel, we converted that energy to dosage based on this process we adapted from our reference paper. So to reiterate, these are not exactly the MC simulations because inherently they're very complex and generally done using software like MC and PX. So we used a lot of approximations. For example, we didn't account for pan angle, we didn't account for geometry of the complex helical CT scan. We did only slice-based photon interactions. We couldn't do physical interactions like per photon photoelectric or Compton scattering or other uncertainties that come along. So these are the results from our MC simulation. This is for fetus 2 and slide 67. You can see the downsampled image and also the relative dose distribution. The actual dose calculated and the normalized dose is very close to benchmark because we use same normalization and scaling factor from the reference paper. And these are some other examples of results for different slices of the same fetus. Again, the doses are close to benchmark, but the main thing is you can see the relative dose distribution for different tissues here. These are our references and our code is also available on GitHub. Thank you.
Great, thank you. Great job. Let me turn up the volume. So I had a question. So can you talk a little bit more how the Monte Carlo simulation was done?
Yeah, so we assumed one rotation of the CT scan, not because it's helical, so it's difficult to incorporate that. Just one scan per slice. Then we consider 10 angles from 360-degree angles. So assume 100 photons are coming from those 10 angles. Then for the attenuation from the tissue, from the periphery to the voxel, we took the value of PW image, that is equivalent diameter for equivalent water diameter. And path length we calculated based on the image positions that are given in the DICOM image. And after that attenuation, we calculated the probability of interaction of the photon with the tissue. And we just took that it is directly proportional to energy of photon and attenuation constant and inversely proportional to the distance. So the inverse square law of X-ray energy basically. And based on that proportion, we calculated number of photons that are actually depositing their energy into that voxel. And after that, we use that method to the method in the paper to multiply it by normalize. Yeah, so we get the mega electron volt per gram per photon from our simulation. We multiplied that with the normalization factor for that scanner given in the reference paper, and then multiplied it by effective milliamperes because we are just doing FTC, not PCM. So effective milliamperes were again given in the data set, and then divided it by CTJ volume per hour.
Okay, great, nice. Maybe you could answer one of those questions.
No, we just did one iteration because even one iteration takes a lot of time. So this is total around 100 photons per voxels, 128 into 128, and then 36 angles. So it's around 58 million photons just for one iteration. So we just did one iteration.
Great. Okay. And then the next question.
They're all watching to the patient and hitting the detector. How is the radiation dose depend? Radiation dose for particular tissue will depend on the distance of the tissue from the source.
The inverse square law?
Yeah.
Just the inverse square law of the intensity, and intensity is also the dose in this case.
Yeah, because X-rays are fan beam, so they are they are distributed in spherical way. At far distance, it will be 1 by r.
Yeah.
Okay.
Yeah, level, there's dose.
1/4.
Great. Professor McVeigh, did you have any questions? I see your. Okay.
I was having internet difficulties, so I just signed on just now. So.
Okay, no worries. You could put it in the chat too if you're having troubles. So great job. So let's move on to the next group. Okay, thanks. Just hold on.
Okay, is that okay?
Yeah, that's good.
Okay. Hi everyone. We're going to talk about the simulation and removal of metal artifacts and CT images. My name is BENG280A.
And I'm Nana. I'm Roshan.
I'm Iris. Okay, so this is the Okay, so the plan for today's presentation is first to go through some background information, and then Nana and Iris are going to talk about the simulation and removal part. So first, what causes metal artifacts? So that could be really dense materials in the body, like bones, metal implants, or dental fillings, etc. And so when you image a patient with those in their body, you can have these behaviors like beam hardening, and they happen basically when a beam goes through a dense material, and then the average energy will increase because the low energy photons are absorbed. And as seen in this graph right here, those beams can cause a darker image in the center, and then the beams that don't pass through the material will have a brighter image. So those will cause like streaks, non-uniformity in areas that are supposed to have uniform colors, and other noises. And then another behavior that can happen is photon starvation. So like the name, it just means when not enough photons are hitting the detectors. So the detectors don't have enough information to produce the image, and this can cause streaks, as you can see in this picture. And then this one has like scratch-like streaks and also noises. And then some methods that are used to remedy these behaviors are filtering and painting, interpolation, projection-based MAR, that's analyzing and modifying the sinogram and doing the projection again. And those will be the things that we go through today. And then some other more novel approaches include photon counting. So that's instead of using like the scintillator to do energy integrating detectors, we use semiconductor materials, and they can register individual photons and calculate the energy individually. Also dual energy. I think other groups presented this using two X-rays with different energies, and then also deep learning. So using neural networks to learn the complex artifact patterns and remove them. Okay, and Nana's going to go over the simulation.
Okay, so in the simulation part, we first focus on the photon starvations. We began with the HU data, which is a House field unit of an image. And we found the X-ray characteristic data table on the right-hand side, and it describes the mu values of water on different energy levels. With the mu values, we can plug it into the formula and convert all the House field unit into the mu data showing in the flow chart. And by setting some of the pixels into a relatively large number, we can simulate an implanted metal in the image. And after that, we will do a forward projection with the radon command to get the sinograms. With the sinogram, we want to simulate the photon starvation. So what we've done is to set a maximum value on the sinograms. And for all of the pixels exceeding the maximum value, we will set it to the maximum value instead of preserving the original values. With the attenuated sinogram, we'll back project it with iRadon and get our simulated image. And as for the beam hardening part, we want to understand like how the mu values in the mu image varies across different energy levels. To realize this, we found a paper describing some beam hardening correction methods, and they introduced like a model describing describing the relationship between like the mu values across different energy levels and the mu values of the water. So with the same, we use like the X-ray characteristic table, and with the model, we are able to generate 120 different mu images across the different energy levels. And with all of the images, we add the metal that we add a metal on it and forward project it into 120 sinograms. With all the sinograms, we add them all together to get like overall sinograms, and the rest of the steps are all the same. We'll set a maximum value and attenuate all of the intensity over the values, and back projected it to get our simulated image. And this is a visualization of all of our simulations. The first image display the mu values, which there are like two T's. We set it to like a a larger value, and it is like the simulation of the implant metals. And the top rows are from monochromatic energy simulations. So in the middle top image, you can see there are like streaks and occlusions, and which are due to the existence of the high density metals. And for all the images in the lower row, they are the simulation of polychromatic energy. You can observe like the last image in the background part. They are supposed to be uniform because they are basically just air, but there are some like uneven texture and streaks in it, and it is the effect of the beam hardening. Yeah, and the next part will cover by Iris.
Okay, so moving on to how did we try to remove the artifacts? We tried two different approaches, or we tried a lot of approaches, but we're going to cover two today. The first one is pretty straightforward. It's interpolation. It's an interpolation-based method, and we do it directly on the sinogram. So we start with the, can you see the mouse? Original image that we just generated, and you can see the artifacts and the teeth. And then using thresholding, we identify the metal regions, so you'll get a mask that sort of just looks like the bright spots of the teeth and black everywhere else. And so then we can do the radon transform of both the original image and the metal region to get these two sinograms. So this region is, where the metal is. And then using the indices of the metal only sinogram, we can interpolate, we can interpolate to get this blurred image on the original sinogram. And then by taking the inverse radon, we get the interpolated image. And you can see there's a little bit more blurring of the artifacts here. And then we did, though, have to replace the metal because the interpolation does remove it. So that was the first approach. And then the second approach was, we're calling it the simplified iterative metal artifact reduction method. And so this was based on a method that Philips uses, but it was hard to find information because it's all proprietary. But the idea is that you want to create this image of the artifacts only with nothing else. And so how you do this is very similar at the start. We threshold to find the metal regions only. We do the transform to get the sinogram. We attenuate the sinogram, and then by doing the inverse transform, we get this image of the simulated metal artifact. And then we subtract this from the original image, and you can see kind of, especially between the teeth, it's reduced a little bit. And so these are kind of all three images put together. We can see in the interpolated image, you get more of a blurred effect, but it sort of introduces some new artifacts, which isn't ideal. And then in the subtracted image, the artifacts are a little bit reduced, but it's best sort of in the center region compared to the original. And so some challenges and future directions. One big challenge was generalizing this method. We did all of our work kind of on 1 to 2 images, all using like dental implants. So if you were to try and apply this to other implants, it might not work as well. And then also we used very specific thresholds. So it was hard to, every time you put a new image in, you would need to sort of modify that. And so that's also a reason why we weren't able to do multiple iterations. You can, keep putting the image in and hopefully get rid of the artifacts over repeating the process multiple times. And so in the future, some things that would be good to work on would be visualizing and comparing the quality of the images with the artifacts removed. Like you can kind of see them in the picture, but it's very qualitative. And so things like measuring the standard deviation, the contrast noise ratio, and the metal artifact noise would be good. And then finally, it would be interesting to also explore deep learning and in-painting methods, but we didn't for time constraints. And here are references. Do we have any questions?
Great, really nice talk. Let's open up for questions.
This was really good. When you put the artifacts into the images, um, you decided what the amplitude of that tooth was, right? And so you just put a pixel mask in there with that, and then used radon to essentially create the sinograms.
Mhm.
And at that point, you know which pixels on the sinogram are metal, right? And you did that with thresholding, is that right? On the in the sinogram space.
Mhm.
I see. So, yeah, and then so the interesting thing is how much that algorithm blurred the other teeth. So if you go back to your pictures,
Results.
yeah, that's the next here. The interpolated image. If you look at the middle teeth, they're all really blurred out. Can you give us a notion how you would deal with that with the interpolated algorithm? Is there a way to change the interpolated algorithm to try and reduce that?
Sorry. We think we actually did the interpolation as part of a more complex method. So we didn't focus too much on it. I guess that was one problem that we were also interested in, because even though you're removing the artifact, you're actually generating more artifacts.
Right.
So that was
right.
we're also
That's
interested
cool.
in looking
Is
at.
this in MATLAB? Do you have this whole process in MATLAB code? And
It's in math.
Yes.
Yeah.
okay. Great. Okay, good. Thanks.
Very nice. Okay, so let's move on to the next one. Great job. Okay. Yeah, they're not talking yet. That's why, Elliot.
Okay. Thank you, everyone.
Okay, can you guys unmute then?
Yes. Good morning, everyone. We're group 12. I'm Gene. This is InQ, and that's JP. And today we will be presenting micro CT on bones. So just a comparative overview. With CT, we're able to analyze and go into the millimeter resolutions, and it's great. It has its purpose for large body scaling, such as if we were to image a human body. However, if we want to get into the microarchitecture of bones, we do need to rely on micrometer as our resolution. So micro CT achieves that with microscale resolution. And here are a side-by-side. We have CT on the left side, micro CT on the right. With the advent of CT in the 70s, it was great. It served us it served our purpose. I mean, we were able to scan humans. However, when we got to the miniaturization and trying to scale it down to the micrometer level, we had to come up with a new technique. So we went ahead and segregated the X-ray generator as well as the X-ray detector. And we also had the rotational element separated, whereas the CT scan has all of them functioning together. Thankfully, that's why the man is probably relaxed and instead of rotating like a rotisserie chicken. I think that's phenomenal for us. And so this is micro CT. Micro CT, we have the X-ray tube, which is the purple. And inside you'll find a cathode and an anode. Generally, the anode is tungsten because it radiates frequency which are sufficient enough to have the photons emit a small percentage of X-ray transmissions. It goes through, and in the middle, we see a rotational stage. The cylinder generally is a sample of 150 millimeters in diameter. So you can't fit much, and most of our studies will be ex vivo because of that. However, it's non-destructive because we have more control on the focal point, and the fan beam is much more concentrated as well as collimation. We're able to kind of improve on those variables because everything's so separated. And once the 2D planar projections, X-ray projections hit the detector, it goes into a computer. And the computer, basically, you can implement algorithms such as Feldkamp-Davis-Kress, which is a great algorithm for 3D model reconstructions. And with the reconstructions, they kind of look like this. So not only can we see through the bone, we could also see inside. And there are two components. The inside, which was the red, is the trabecular bone. The outside is a cortical. And these 3D models help us in early detection of osteoporosis, the development of grafts and implants, as well as teaching anatomy and pathology. And that third component of anatomy and pathology, we want to elaborate on. And we want to see how micro CT is able to investigate the effects of type 2 diabetes in mouse models. So we'll be looking at femurs, bones, and inside the trabecular structures. And now I'll pass it over to JP, who will be talking about bones.
Thank you, Jen. So we're going to apply this technology to bones. Just as a reminder, as Jan mentioned, there are two main components in this multi-level structure called bone. One is the compact bone or cortical bone, which accounts for around 80% of the bone mass. It's hard and dense. And also we have the spongy or cancellous bone, which is less dense and accounts for the 20 other percent. It's important for this project to understand and to know how the bone modeling process works. So there are mainly three cells involved in this process. One is responsible for the resorption, called osteoclast. One is responsible for the formation of the bone, called osteoblast. And a derived cell, a third type formed from the osteoblast, called osteocytes, accounts for most of the cells present in bone, 90 to 95%. They are the ones that regulate the whole process, connecting osteoclast and osteoblast, among other functions like regulating the mineral density of the bone, as well as sensing the mechanical and responding to the mechanical stresses. So where do these osteocytes, these coordinated cells live? They live in very, very tiny holes called lacunae that we're going to see in our micro CT images, and they're connected through small canals called canaliculi. I want you to focus to pay attention into this small black dots, which are actually the lacunae, so where the osteocytes live. In this project, we analyzed a bone from a controlled healthy mouse and a diabetic mouse to compare how many of these small porous where osteocytes live are and how big they are.
So now we perform segmentation to quantitatively analyze the difference between diabetic and control bones. As we learned in this lecture, bone absorbs more radiation, resulting in higher intensity values. So we will use this property to segment the bone first. We are going to use Otus method. It is an adaptive threshold method that determines the optimal threshold value by maximizing the between class variance in the histogram. So the right-hand side is our histogram. And if we naively just apply our Otus method, we get the background. So after we have to remove this peak value from the background, and then we get our rough mask. But this is not really good enough because we still have some outliers and holes. And this time, we are going to use a convolution operation or morphological operation. Let's say we convolve our 3x3 clonal on this image, and we assign a value of 1 if every ones in kernel align with every ones in the image. And it's called heat. Or you could think of it as everything in gray is 1, then we assign a value of 1. So everything here is 1, so we assign a 1. Same here. But at the top, in this case, we have a 0, so we assign 0. And same here. So we just repeat it, and you get the idea. And there is another operation called fit. We assign a value of 1 if at least there is one value is 1, then we assign 1. So at least 1 is 1, so we assign 1. But here, even though we have 0 here, there is at least one value is 1, so we assign 1. So in the end, we get all ones in the fit operation. And this is our original image, and this is the result after heat, and this is the result after fit. So we call this erosion, and we call this dilation. So dilation is used to make the image thicker, and erosion operation is used to make the image thinner. So going back to our original problem, we had lots of noises here. So we first apply our erosion to remove the outliers and make the image thinner, but we want to make it thicker again. So we apply dilation. And this erosion followed by a dilation is called opening. And closing is the vice versa. We fill the holes and then make it thinner again. So closing is dilation followed by an erosion. So now we have nice looking bone, and then we want to segment the lacunae from this bone. This time we use Otus method again, but this time we select the left-hand side, which is more darker region, and we compute the union of this darker region with our bone mask. Then we get our lacunae and bone mask. So top side is our control bone sample, and bottom is our diabetic samples. And we move on to our quantitative analysis using these two masks.
Thank you. So the results we obtained were that the lacuna density, so the number of lacunae, remember these pores or holes where the osteocytes, the coordinated cells live, were smaller for the diabetic bone, as well as the lacunae percentage, so the space they use in the bone, are smaller for the diabetic bone. This means we have more pores in the control bone, which can be counterintuitive. However, this aligns with literature. For example, this paper by Woolley et al. Shows that in diabetic bone, they both find less lacuna density and less lacunae volume for diabetic bones. It's also interesting to note that the crack growth toughness in the diabetic bone is way less. This can be counterintuitive because a material with more holes, how can it be that it has less crack growth, right? But it actually makes sense if we consider the bone health and not only the amount of bone that there is within it. So, for example, the bone remodeling process, because as we have less osteocytes, it is affected. The reduced blood flow that we get with the less connections and worse network, and the impact on other regions of the bone, for example, the mineral region. And here are some references that we use to build this project. Thank you very much.
Great, nice job. This is now open for questions. Any questions from out there?
I have a quick question. Did you estimate what the actual resolution is of the CT images you've got, and the distribution of holes that you might miss because your resolution isn't high enough? Number one. Let's just do that question first.
What was the question again? I couldn't hear the first
What was
part.
the resolution of your CT, and do you think you might have missed some of the lacuna because of your resolution wasn't sufficient?
The resolution of the CT was 1.6 micrometer,
Okay.
and I don't think we missed lots of lacunae, but we have So like at the end, we use the particle filter because we know the size of the lacunae, the volume of the lacunae. It's between 50 micro cubic meter to 200 cubic micrometer. So we use that filter to get the final lacunae mask, but like 1.6 micrometer was good enough to get the like lacunae.
Okay, great. Did you have another question there, Elliot?
No, that's good.
Okay.
Thanks.
So yeah, so there's want to answer that question from the chat? The last one there?
So one factor that played a huge part in our presentation and project was the quantification part, which InQ spoke about. When we attempt to isolate and segment these bones, there is a process called Otsu thresholding. And at this point in the future, we foresee this becoming more advanced, and AI particularly is also playing a big part in this. So we don't see much challenges, but we do see a little bit more aid from computer systems being able to diagnose once we get to the in vivo side of things. Because the magnitude of magnetization is much bit larger. When you move, things just go out of hand. So we need to work on maintaining a static sample, and anesthetization might play a huge part in this.
Okay, great.
We
Nice
have one-
job. Yeah, really quick. Go ahead. So the question was, did they consider a fractal analysis?
Yeah, we did not. I think it's a very good idea. Thank you for the suggestion.
Okay, great. Thanks a lot. And let's go to the next group. And if there's questions on the chat that you didn't answer, just your group just answer them on the chat. Okay. Okay. Guys ready? So I'll mute and you guys can unmute, okay?
All right. Hi, everybody. We're excited to be talking to you a bit about beam hardening today. My name is Ariana.
My name is Ria.
And I'm Anirudh.
So we'll take you through some background on beam hardening first, and then touch on the artifacts we can observe in beam hardening, and lastly, dive into the simulation that allows to reduce the artifacts. Now, we touched briefly on beam hardening already today, but I'm still going to provide you some more details on it. So it describes the phenomenon that the mean energy of an X-ray beam while it passes through material increases. Why is that the case? Well, we already learned in class that the attenuation coefficient does depend on the energy, right? The graph here on the right shows the attenuation coefficient mu for bone, muscle, soft tissue. It's much lower for higher energies. So this, for polychromatic X-ray beams, does result in a, of course, slightly differentiated spectrum we will measure. Low energy photons are going to be attenuated more than high energy photons. This gets specifically tricky when we look at non-homogeneous samples with different attenuation coefficient across the object. In order to understand a little better, how does it actually affect our spectrum of the X-ray beam and then our measurement, let's look at those two graphs. On the left, we can see an X-ray beam that left the tube in blue, and the same X-ray beam that already passed through some material in red. The one that passed through some material, I think it's pretty straightforward. You can see it's pretty much right shifted, higher energies. The lower energies are not even there anymore. So what does this mean? Well, if it again passes through material, the intensity we will measure then is going to be higher than it usually should be because it's being less attenuated. This is what you see on the graph on the right. As reconstructed means this is what we've been measuring. Corrected is the curve that is accounted for the beam hardening and is slightly below the detected one. Now looking at the math, we'll make a little more clear where we actually do get the beam hardening into our reconstruction. I think for monoenergetic, just to take a step back again, I think for monoenergetic, we do understand how to calculate the intensity, right? We've seen that formula for homogeneous sample multiple times. For non-homogeneous, we're gonna integrate over dx for mu. That's also clear, I think. Now, where we do see the beam hardening is for polyenergetic sources. And we can calculate that intensity by integrating over E. I think that's also over the energy. That's also understood already from class. Now, for a non-homogeneous sample, we would have to integrate mu also over X, right? Which is actually not applicable in practice because it makes it very complicated and we don't know the coefficient across our entire sample. That's why we use this formula in praxis and account for the beam hardening with polynomial functions to do the correction. Annie's going to talk about this in a little, but now I'm going to hand over to Ria first.
Yeah, so we're going to talk about two artifacts that we typically see from beam hardening. Both of these also kind of have been touched on earlier today too, so this is just another refresher for everyone. The two are cupping and streaking. So cupping is when the edges of an object appear brighter than the center, and streaking is when you can see dark streaks throughout the image. So we're going to talk about cupping first. It's best visualized seen through an X-ray through a uniform cylinder. So on your left, you're going to see two images, one of them with beam hardening artifacts and one without. So what basically happens is as X-ray pass through the middle portion of the uniform phantom, they are hardened more than when they pass through path A, which is through less material. So when they are hardened a bit more, their attenuation coefficient is going to decrease, resulting in the measured value being shorter than ideal. So we can see that actually when we put a line through the projected image and then plot that line, and you can see that in C and D figures. The one on the left, which is the beam hardened artifact, you can see a slight dip and it come back up, and that's kind of where we get the idea of what cupping is and where the term cupping came from. And this is the attenuation profile, so basically the CT number plotted across that red line that we just talked about. The next is called streaking. Streaking, as you can see on the right image, is a very small, I think it might be hard to see, but it's a small darker line that goes through this image. And then on the left side, you can see the streaking throughout the entire image as the CT goes through the body. So basically, streaking is another common artifact found, and it's common between two bones or two highly dense materials. This is a portion, what happens is a portion of a beam when it passes through the first material at a certain position is hardened more than when it passed through both objects or like another material that where it's hardened less. So at certain positions, the beam is hardened more than at other positions, which causes streaking or that darkening as we see even in cupping to occur in a line. As I said, this is common in like a lot of bony regions or when contrast is used. So now we can talk a bit about methods to prevent artifacts. We're going to go through a simulation after this, but we also have a couple methods like dual energy scanning, which was talked about on Monday actually, where we can use two different X-ray levels to capture an object at two different intensities. This is used to identify specific attenuation patterns of materials and characterize the tissue, and then we can correct for the uneven attenuation after. You can also use some filters to make the beam more uniform as it passes through the body. So pre-patient filters can absorb soft X-rays, and bow tie filters can potentially pre-harden X-rays.
Yeah. Okay. So now we're going to talk about the simulations that we used for the reduction of beam hardening and for the simulation of the beam hardening itself. So first, we used the method we used is called iterative reconstruction. So iterative reconstruction is an algorithm that involves refining the image through multiple iterations of like estimation and correction. So here, the raw data from the CT scan measurement is taken, and it is compared to a modeled or like a forward projected image. which is like an initial guess of what the ideal reconstruction should look like. It would be like the sinogram, like a forward projection sinogram of the ideal image. And that is then compared to the raw data that we have measured from the CT scan. And the difference is then multiplied with the learning factor and then performed over multiple iterations to reduce the error with each iteration. We can see right here, this is actually a highly simplified model of how cupping would appear. Obviously, we used a phantom, which has, so we know the ground truth for the image, and we simulated the cupping artifact here, and you can see that in the graph as well. So for the correction itself, this would be the corrected image with 10 iterations of the iterative reconstruction algorithm. And if you see the graph on the right, then you can it's quite faded, honestly, but then you can see that the faded portion is how the original central intensity profile looked like for the image that we had with the cupping artifact. And with each iteration, the central profile gets closer to the original image. So with each iteration, the error is reduced. And we have done the same thing as well for streaking. Streaking, you can clearly see in this case, I don't think it's quite visible up there on the screen, but essentially between the two images right there, yeah, it's quite faded on the screen right here, but then between the two images right there, you'll have dark streaking bands, which can be observed on the right side graph as lowering of the energy profile between them. And at the same time, you can also notice that on the high density objects themselves, there's also cupping artifact observed in the image. So they kind of go hand in hand and they occur together in the image. And similarly, we have the corrected image here with the 10 iterations of the algorithm. And with each iteration, the error reduces and gets closer to the original image. So there's also another method that Ariana touched upon earlier, which is polynomial fitting, which is more applicable to the energetic model, where the relationship between the attenuation coefficient and the energy and distance is simplified using a polynomial function. And that is then used to add to the forward projection of the image the data that is collected from the detectors and then applied there to get the corrected reconstruction. But the method we went for was this, because we use phantoms and it's more straightforward this way for the phantoms. And with that, we've seen what beam hardening is, how it affects the CT imaging, as well as some of the methods we can use to reduce them. So we'll open the ground for questions.
Great. Very nice. So I got a little lost on, so what was the cost function for your iterative reconstruction?
So in this case, because we use phantoms, we already know the ground truth. So in this specific case, what I did was it just compares the forward projection, that is the sinogram in this case for the ideal image, with the forward projection or sinogram for the image with the cupping in it. It takes the difference of the sinograms to find what the error is and multiplies that with the learning factors to slowly improve upon the the reconstruction of the sinogram of the image. No, the image with the artifact, and then it gets closer to the original.
But how would you use this in practice? Because you don't know what the ground truth is.
For that, we use, like I mentioned earlier, that we use something called model data, like a forward projected data, which was used for like actual image, like I guess actual CT images, right? So that is done by modeling them with the attenuation coefficients and of known tissues and like guessing how the image would be perceived, I guess, without any noise or without any artifacts. And since we use the phantom, we just directly use since we know the ground truth already, we were able to use that, but then for actual imaging, it would be with an initial model guess.
Okay, so if you had a human body, what would be your initial model guess?
I guess, okay, so that would be built upon depending on what you are imaging, they would make guesses based on the attenuation coefficients known for the known tissues that are present in that scan.
Okay, great. There's a question online. You could answer that.
Yeah, so the learning factor would be important in case of the initial guess that we have, because the initial guess that we have wouldn't be exactly accurate. In case of phantoms, we know the exact difference, but in case of the actual imaging, we wouldn't know the exact how the image would exactly look. So that's why we use a learning factor so that we can change it according to whether or not it gets closer to what we would like the image to look like.
Great. Okay. If there's no more questions, great job, and we'll move on. Thanks.
Thank you.
So next group, come on up. Okay, go ahead. I'll mute and you guys can.
Hello, everyone. My name is Caitlin,
I'm Wesley. And I'm Carter.
and we did our project on the effects of filter choice on noise and signal detection in CT imaging. So before we get started, I'm just going to go over a really brief overview of CT imaging. To refresh our memories, CT imaging converts multiple X-ray projections into a 3D image, allowing for internal body visualization with high precision. And filtered back projection is a widely used algorithm in CT reconstruction that helps transform 2D projection data into a clear cross-sectional image by applying mathematical filters. So for this project, our group was aiming to understand how different filters used in filter back projection affect the characteristics of noise in CT imaging.
So to start off our project, we wanted to make like a skeleton just to make sure we're on the right track. So a part of our code was put here. We used Gaussian and Laplacian just to get a feel of like what the image is supposed to be. We used the abdomen picture, and we also did radon transforming as shown in the graph. But essentially throughout the skeleton code, we wanted to use InRead in MATLAB to read the image and grayscale it in general, so it could apply to any image, even if it isn't grayed already. And we used M2 to improve the accuracy and filter for the next filtering things that we did to be similar to the stuff we got taught in class.
So here's a quick little visualization of the filters we ended up playing around with in MATLAB. So this graph you see is a frequency response graph where the x-axis is the normalized frequency and the y-axis is the amplitude, which indicates how each filter attenuates or preserves specific frequencies. So we did a range of filters, including low pass, high pass, and band pass. On the low pass side, we have the Hanning and Hamming filters. On the high pass, we have the Ramlach and Shep-Logan filters. And we also included a very simple bandpass filter.
So we ran 3 specific experiments. The first one was with the traditional ramp filter. And we wanted to understand how if we cut off specific frequencies, like we just mentioned before, how that's going to impact our image. So what this simulation is showing you is as the ramp increases or as we cover more of the frequencies in our specific image, you can see that the image will change to kind of go from a blurry mess to a nice clean image, and then the noise will start to fight back towards the end. And that is something that we applied to an abdomen image for the sake of our study. Then we also tried this with the Shep-Logan and some other filters, but the main one that I'm showing right now is the Shep-Logan filter, which has a kind of tapered off tail to it. So the amplitude at the end does not go up to 1 as it does in the ramp filter. And so you can see similar characteristic traits being applied to this. However, at that higher noise, at the higher noise frequencies, or at the higher frequencies, excuse me, not the higher noise frequencies. At the higher frequencies, you can kind of see that the image doesn't fade back as much. We can kind of see our original image a little bit. And then this is the one that I thought, or we thought was kind of the most interesting, a bandpass filter. So this isn't really changing the actual amplitude at all, but essentially just cutting out frequencies from distinct locations. And you can see that this creates really interesting artifacts. So on the left, we essentially have a circle that's like growing out, and you can kind of see more and more of the image while keeping that traditional circle kind of there throughout the whole thing. And then on the right, when you start to shift this bandpass filter at these different frequencies, you start to see the circle kind of go from the bottom to the top instead of from inside and out. And obviously that has a drastic impact on how much of the image you can see and what parts of the image you can see.
So overall, based on what we saw, at least, the images are kind of small, but the high pass and low pass worked like we expected. High pass seems to start to grow in sharpness until the higher frequency was chosen, and the low pass started to like gradually smooth out the image even more around the edges, and it would become more blurry. And lastly, the band pass seems to be like the most strange in that we think that the circle artifact is like the focus point where it's like over blurred, and then it starts to try to sharpen out and combine the two. So at least from the results, it is what we expected from all three types of filters applied. Oh, so for the future directions, we want to explore other filters, of course, like see how various different types of low pass filters would affect the image. Like, I mean, like the ones we learned in class, there's different types of low pass ones. We also want to potentially add automated system that could just apply a frequency that's good already, just to help with the ease. And we also want to apply more clinical data. Like we don't just want to apply it to the abdomen picture, we could apply to the brain, for example, over here. So if we could do various different types of data, various different types of images and different filters, then I feel like we could gain a lot more from this type of project. And our references are here. Thank you very much.
Great. So nice job. Could you explain a little bit? I got a little confused. So which domain are you actually doing this in, the frequency domain, or?
Yes. So this is being applied at the iRadon step. So if you, MATLAB traditionally has like a function that you can at the end add on like what type of filter you want to apply. So it's applying it in the frequency domain when it's flipping it back.
Okay, so you're letting iRadon do the
yes, yes. Okay. We had to, for the bandpass filter, work a little bit or do a little more work because there is no traditional bandpass filter setting in MATLAB. Obviously, the iteration was more of a challenge for our group getting the movie to kind of show how it's changing at these cutoff frequencies. But for the bandpass, we actually had to directly do the convolution and show how that changes. So that one was slightly different, but for the main filters that we looked at, it was at the iRadon.
I see. So you can involve the projections with the basically the impulse response of the bandpass filter.
Exactly. That's correct.
Okay. Cool. Any other questions?
What does the
go ahead.
What does the bandpass? Sorry, you okay?
Go ahead.
What does the bandpass filter represent physically? Like, how would that happen in when you were scanning?
I think like many different filters, it varies in usage. For ours, it clearly wasn't very great, but I mean, the bandpass overall wants to balance both high and low such that we don't get the over noisy downside of the high pass and the over blurred aspect of the low pass.
I think we took it from a more theoretical standpoint, if I'm being completely honest. Realistically, like you just mentioned a ramp filter, those are pretty well defined in terms of physical space, but having something that essentially just cuts off specific frequencies, especially is quite challenging because it essentially means it's completely getting rid of frequencies.
Right.
Which
Right.
that I think it theoretically is why we try to represent it that way. But maybe fundamentally or physically in the lab, it's kind of hard to say.
Okay. Great.
Weird if they your image like move around.
Yeah, it was definitely interesting, for sure.
It definitely showed that not all images like could be used on certain filters. Like the person using the actual system has to choose and decide for themselves what they want.
And also design the filter itself, right? You can't just put any filter on the machine or on the frequency domain and say, hey, this works for our specific image. You have to kind of take a step back and maybe understand fundamentally what that filter is going to do, which I think the bandpass shows extremely well that maybe that isn't a great decision to really go forward. And that might be why in the lab we don't have anything physically representing that yet.
Oh, okay.
Great, thanks.
Awesome. Thanks.
So let's do, I think, our last presentation of the day, right? Okay.
Yes.
Should get out of here on time. Okay.
Good job. And do an air one.
We'll do it a little bit.
Yeah. Hold on. Give me one just a few.
Let's check the discussion section. Nobody.
Thanks for covering.
No worries. I think that makes sense.
Hello, we are group 16.
Hold on, make sure you're unmuted. Are you unmuted?
I think so. Daniel, and this is our project 1 regarding CT. So our group was tasked with creating a simulation in MATLAB to investigate the spatial resolution of a CT image as a function position in the field of view. So before we get into the results and our methodology, I want to give you a bit of background information. So as you guys are all familiar with, the first thing we tried to discover was the radon transform, which is in the 2-dimension function is a set of line integrals. This transform computes the line integrals from multiple sources along parallel paths as shown in the picture, and these are called beams in a certain direction. And to construct, to represent the full image, it takes multiple parallel beam projections by rotating different angles and rotating the source around the center of the image. And then projections are able to be computed by any general angle theta. And as we did in our homeworks, we can split up the projections in the x and y axis separately. Although the radon transform can be thought of as a general mathematical approach that can be applied to many projection geometries, we chose to explore one in particular called fan beam geometry. It is somewhat similar to the radon transform in that it is also has projections of 2-dimensional functions as a set of line integrals, but instead of having multiple parallel paths, it emanates from a singular source, and we take the line integrals from the paths from that singular source. Yeah, it is similar to the radon transform in that fact, but except it includes additional math regarding the transformation.
I'm going to talk a little bit about the theory about why the resolution in a CT scan is different across the field of view. So up top, we have our filtered back projection equation, our inverse radon transform with the filter WQ and with the X-ray blurring SQ. And the reason why we get blurring is when we compute this integral in practice, we take discrete samples of the integral and then we multiply by the delta factor. So when we can't actually compute this continuously. So our samples across dq, which is our radius or distance from the center, and d theta, we get unevenly spaced samples like this. So when we try to compute our point spread function, which is our way of characterizing the impulse response of the imaging system and our way of characterizing the resolution of the system, we get different impulse responses based on the location in the field of view. So if we take a look at close to the center of the field of view, these our samples are very close together, which means that if we have an impulse in near the center, our impulse response will be a rect function or a Gaussian function with a very small window, which means which allows more of the high frequency components of the signal to pass through. However, once we get to the edges of the field of view, each sample is very far away. So if we have an impulse at this location, our rect function will be much wider, and that results in a low pass filter on the outside. So basically what that means is there's high frequency information close to the edges of the scan can be lost. Another issue is increased interpolation. So when we interpolate between these points that are very far apart, we're adding a lot of noise that is not represented in the actual signal. So what this means for the expected result of our experiment is that our point spread function will be wider as we get further from the center of the field of view, and we would get more aliasing and more of a low pass effect closer to the sides of the image.
Yep. So I'll just go quickly about our MATLAB simulation of this spatial resolution in the field of view. So we took our the method we used last in the homework, and we add evenly spaced impulses. We first create a 256 by 250 matrix, and we evenly space the impulses at 32 pixels. And we apply the radon transform, and we get the sinogram. And then we reconstruct the image using inverse radon transform. And the parameters on the top left, top right, which we just use angle from 0 through 180 and use the delta theta at 1, and we just use a RAM lock filter to do the inverse radon transform. And we can see at the surface plot where the height represents intensity and the XY plane shows spatial position. To quantify the spatial resolution in the field of view, we look at the impulse with the point square function and full width half max. We first look at the center of the field of view, which is at 128 x at 121 at y at 128, and we cross the line and across the axis at y-axis, and we look at the point square function where you can see kind of the center has the most spiky signal, and to quantify that we calculate the full width half max, and we can see at the center where the full width half max is the minimum, which means the spatial resolution is the optimal. And we did the same thing to a peripheral impulse at 3232, and we can see the signal is averaged out without like a spike, and the full width half max is the range is pretty small from 145 to 165. So that may be some like artifacts, so why it's not like even, but it means that at peripheral impulses the resolution is less than the center. And we did the same thing to the fan beam transform, and we did this. We reconstruct the image. We first transformed the image we got using fan beam transform, and we reconstruct the image using the inverse fan beam transform, and we get this plot where the center is pretty bright. And we did the same thing. Look at point square function and full width half max. Here we are not fully understanding why is it the case that the center is not the minimum, but it's like all of them are pretty large a number at the full width half max. So yeah, we're still working on that. Same thing with the peripheral impulses and the future.
Some of our future work would include like incorporating an X-ray detector blur, which accounts for quantizing the number of samples across a wide detector. So that would also add like a sort of low pass effect where it spreads out the point spread function, and it would be interesting to see how that cascades through the system, and how the radial effects of resolution are shown like with a more noisy signal. Another thing we would want to test is like more impulses across a wider image. So our for the sake of our computational complexity, we had a smaller image and a fewer impulses, and we can maybe characterize or find a better fitting equation if we have more impulses. Another thing we can do is reduce the number of samples. So we had data going from 0 to 180, and we could reduce that step size, and that would actually increase the chance for undersampling at the edges. So that would mean more aliasing and more of a blurring at the edges of the image. And another thing we wanted to take a look at is using like a metric other than full width half maximum to characterize the resolution at each of the impulses. So the reason why we wanted to look into that is the full width half maximum, or even the point spread function on the x-axis and the y-axis, doesn't tell the full story of the blurring. If you closely observe some of the artifacts along the edge of the image, you will see like a sort of like radial blurring where it sort of takes the shape of a multivariate Gaussian distribution, but it is like stretched in almost like radial circles around the center of the image. So to like measure the area of those pulses, or characterize the shape of those, would also be a good thing to look at in our experiment.
And here are our references here.
Thank you. So do you think there's any, I mean, you looked at a grid of impulses, and is there any chance there might be interference between them? Did you try just putting one impulse at different locations, and did that give different results?
I didn't do that, but I think we did that at the last homework. And I think the results are kind of the same. And yeah, I think you're right. Maybe there's some like inferences that we should care about.
Okay. Let's see. I'm not seeing any questions online. So anyone in the room? Okay, well, I think we're right on time. So thank you again. And that ends today's lecture, and Monday's a holiday. So if you come here, there won't be anyone here. I won't be here. So we'll see you next Wednesday. Okay, thanks.
Thanks, everyone.